Okay, good evening, everyone. It's my pleasure to introduce Mélodie Ambroset, who is going to talk about how to publish reproducible image data processing and analysis workflows. Mélodie is a third-year Ph.D. student in INCIA, and she has a bachelor's in cognitive psychology and master's in neuroscience. So over to Mélodie.
So hello, everyone. Thank you for joining. Okay. I checked that everybody can hear us. So I'm happy today to present how to publish a reproducible image data processing and analysis workflow. And I'm gonna lean on the article of Kota Miura and Simon Nørrelykke, probably mangled that name, sorry, and which is called Reproducible Image Handling and Analysis. And as a brief outline, we're going to talk about the fact that digital images are data, that they come with a set of ethical guidelines that I'm going to remember, and that this data can be compromised by inappropriate manipulation. I'm going to define mishandling and misconduct. I'm going to talk about causes of mishandling, and I'm going to talk about the author's proposed solution to this. And the solution is reporting reproducible image data processing and analysis workflows. So I'm also going to remind everyone what is a workflow in bioimage analysis. And I'm going to describe what is a workflow package, which comprises code, description of the code, and the original image data. And finally, I'm going to briefly talk about one recommendation for editors that the authors make and that I really like. And I'll give my references and a set of cool resources for people who want to know more about this. So first of all, I want to remind that digital images are data. So what does it mean? It means that digital images are numerically sampled data that represent the state of a specific sample, for example, for us, frame slides, when examined with a specific instrument. And in our case, often it's any sort of microscope. And so it could look like the image you see on the right, which is an image from one of our experiments. And you see that it's not necessarily a pretty image, even though it's beautiful in itself. Its vocation is not to be pretty, it's to represent the state of this specific sample, which is my brain region highlighted in purple there. And what it's not is artwork or pictures that needs to be beautified, as this pretty digital artwork neuron here. So it means that this data comes with ethical guidelines for the appropriate use and manipulation of scientific digital images. And those have been, for example, defined by Chromie in 2010, and I want to briefly tell them. So the first one is that scientific digital images are data that can be compromised by inappropriate manipulation. Manipulation of digital images should only be performed on a copy of the unprocessed image data file. You should always keep the original data file safe and unchanged. Simple adjustments to the entire image are usually acceptable. It's not bad to process your images. You just have to be careful. Cropping an image is usually acceptable as well. I mean, usually, if you're not trying to hide important information on purpose. Digital images that will be compared to one another should be acquired under identical conditions, and any post-acquisition image processing should also be identical from one image to another. Local manipulations that are specific to one area of an image and are not performed on other areas are questionable at best. The use of software filters to improve image quality is usually not recommended for biological images. Again, we're not there to beautify them. Cloning or copying objects into a digital image from other parts of the same image or from a different image is very, very questionable. Do not do that. Intensity measurements should be performed on uniformly processed image data, and the data should be calibrated to a known standard. I'm not going to expand on that, but just know that measuring fluorescence intensity on images is extremely hard to do well. You should also avoid the use of lossy compression. You should be aware that magnification and resolution are very important. And you should be careful when you change the size in pixels of of a digital image, because if you're not careful, you could deteriorate it because of interpolation. So this data can be compromised by inappropriate manipulation that do not respect those ethical guidelines. And I'm gonna give you the definition of the author's definition of mishandling and misconduct. Mishandling is the wrong handling of image data in general, including analysis and figure creation. And mishandling can be accidental or intentional. And misconduct is when researchers publish scientifically wrong results, regardless of the cause being accidental or intentional. And the difference is misconduct is the public affair. Publication of mishandled image data is misconduct. It's a public affair because it concerns the author, the editor, the people who are going to read the article and are gonna be misled by these mishandled images. So some example quickly of image mishandling. For example, the use of lossy compression, a classic. In A and B, I don't know if you can see my cursor on the console. You can see in A and B that two images that look fairly similar, but one is the original raw data image, and one has been successively saved as a JPEG five times. And you can see in the zoomed in square here in C and D that actually doing this causes damage to the image and provokes a loss of information there. So this is very telling, you're losing information. And another example is a local contrast enhancement. In A, you have an original image of two nuclei. You can see that one is kind of brighter than the other. And in B, you have an appropriate form of contrast enhancement, where the contrast is enhanced, but the difference between fluorescence intensities is preserved. And you see you have a totally wrong way of manipulating your image, which is enhancing the contrast for each nuclei separately. So what are the causes of mishandling? The first one, of course, is fraud. So I'm going to skip that one. We all know why people do that and why it's unethical. But also, in a good number of cases, it's because of lack of knowledge and because also of the apparent ease of use of softwares for novice users. Before digital images, we had dark rooms and specialists, so it was fairly, it's fair to say that these people knew what they were doing to their images. It required a certain amount of expertise. And then in the 90s, Adobe Photoshop appeared. Can you admire this beautiful fresh logo of Photoshop? And the problem is it was accessible to anyone, basically, without any kind of expertise. And now it's even worse because we have a huge amount of softwares that have an easy one-click route to unwittingly alter images, as Aaron and Shu say, especially commercial softwares where you can click on one icon and detected and your objects are segmented and you have no clue what kind of algorithms have been used to do that to your image, and you don't know where your pixels are at and how they've been altered or not. And another thing I want to show you is this answer to a survey question from Miura's team in 2015, and they've asked people which resource they missed the most for optimally analyzing images, and you can see that no matter the background of the people, what they missed the most, at around almost 70%, is knowledge, actually. So support from specialists and courses about image analysis. So what can we do to prevent mishandling and misconduct? Since there is no one-size-fits-all precise set of rules or guidelines for editors to implement and authors to follow, a solution is, as proposed by the authors, reporting methods in a way that renders the experiments reproducible. And to do this, it's the publication of the workflow package that I'm going to describe later. This is a sort of methods reproducibility, which is the original meaning of the term reproducibility, and which is defined as the ability to obtain the same exact results by implementing procedures using the same data and the same tools. And what's interesting about this sort of reproducibility is that it's the only one that can be verified before publication, so kind of before any harm is done. But what I also want to emphasize is that reporting methods in bioimage analysis is important for reproducibility, and it's essential, but it's also sometimes very useful and necessary to understand conflicting biological results. And here illustrated, you have an image, fluorescent image of bacterial colonies. And on the right, you can see that it was given to two different experimenters with the exact same set of processing steps, except that these two experimenters use different parameters for these processing steps. And you can see that the experimenter on the right finds that the area covered by this bacteria is 4 times higher than that for the researcher on the left. And this is perfectly fine, actually, if you report your parameters and the steps that you use, and that you use them consistently throughout all of your experiments, and that your images are acquired in the same conditions, then it's fine. You know, what is not fine is comparing experiments that have been analyzed by different people with different parameters. So, about this solution proposed by the authors, I just want to remind you or explain for those of you who have never done image analysis, what is a workflow? Bioimage analysis workflow is defined as a set of computational tools assembled in a specific order to process bioimages and estimate some parameters relevant to the biological system under study. And usually it looks something like that, roughly. It can vary a little bit, but you have the acquisition of the images. You have some kind of pre-processing steps that are there or not, for example, stitching tiled images or re-registering images, stuff like that. And then you have processing steps, which consist of signal enhancement. You want to get the better signal to noise ratio possible, and you want to do feature detection, which is like you want to be able to extract objects of interest from the background of the image, and segmentation. And when you have done all of that, you have the actual analysis of morphometric parameters and whatever you have extracted out of these images. So now the workflow package that the authors want us to publish to be reproducible consists of 3 elements: the workflow code, the workflow description, and the original image data. For the workflow code, you have different softwares that support reproducibility a little bit differently. Some softwares have the highest degree of reproducibility, and those are widespread. They are free. They have GUIs that allow the recording of your workflow, and they have full scripting capability in 1 or more common programming languages. And example softwares are Chili, Self-Profiler, QPath, or for the more programmers of us, the Python or R. Other software get a good effort mention, but they're not perfect, and they are those who are locally common, who are commercial, like they require paid license or full access, and they are scriptable, but sometimes in proprietary or local language. And for example, it can be Imaris that is used to do 3D imaging, or MATLAB, which is widely used. And some of them are not or almost not supportive of reproducibility because they are rare, commercial, or not scriptable, which means they're GUI only. And examples are Photoshop or PowerPoint, or even undocumented code living on one person's computer. And I'm gonna give you the example of the macro recorder in ImageJ, which makes it a very cool and reproducible software. And in ImageJ, you can basically record every click that you do in the GUI and every step that you perform on one image. And it's very easy to turn this specific set of sequential actions and turn it into a commented code that documents your workflow and that can be reused later. For Python or MATLAB, it's as simple as turning your comment history into a script. So why do we write code for bioimage analysis? The first reason is for automating tasks. Like it's especially important now that image datasets are so big. Like who wants to count cells, thousands of cells by hand, right? And it's also important to separate everything that has to be done manually, such as drawing of regions of interest or final verification, from everything that can be done automatically and reproducibly by the computer. You save weeks of effort. If your computer takes 1 hour to do one image and you have 200, you want it to do all the computing in one go. And retrieve the analyzed data afterwards. But what I want to emphasize is that programming is also a form of documentation of a workflow. And a code ensures that the workflow is reproducible, that the same results can be obtained by others, peers and reviewers, which makes it essential even if it's you only used once and it's not going to be reused by anyone else. It's also in itself a good form of documentation of what you have done to obtain these results. And just an advice, you can keep it simple. A plain text file is good for storing code. Now, code is not sufficient for reproducibility, and it also needs a description. And you need to describe how to reproduce your results. So code needs a metacode, or it's a fancy word to say that a text describing the outline of the workflow. And this metacode needs to explain the overall aim. And here is an example of taken from my GitHub and a workflow I put there. It also needs to explain the outline of the workflow and its key components. For example, a brief description of the 4 steps that my workflow does. Or a more in-depth description of specific processing steps that it does to images. It can look like that, like a text, but it can also look like a flowchart. It's just as informative. And it needs to have instruction on how to run the code, for example, how to install it and how to use it. And it needs to provide download links for the software that you use and any plugins that you need to run the code with name, version number, and location. And usually a URL is sufficient for that. What it also needs is a tables of parameters that you use to obtain your results. And for me, I like to store them in text files as variables that the software can retrieve later. But I'm also paranoid, and I like the macro to register a summary of what all the parameters used were for a given analysis. So it looks like the second image. But any form of text file or XML or JSON file is okay for those, just as long as it can be read by a simple text editor, it's good. The description should also have details about the original data, like the biological name of the sample, the type of table if you used any, the space and time resolution, and a download link for the data, which brings me to the third element of a workflow package, which is the original image data. And the question you might want to ask is, do we need to publish all of it? First of all, I want to mention that publishing source data is a relatively modern practice that came with open access softwares and data storage tools. It was not very common, and especially not for image data. But in any case, data published should be raw data in the format that it was acquired. Usually it's diff for microscopes or proprietary formats. It should come with an appropriate description, and it should come as a structured folders inside a compressed folder or a data container, such as a zip file. I want to mention also that data publishing for reproducibility is different from data archiving, and by that I mean it does not have to be the whole data set, unless you intend this data set for reuse by others. And basically, you don't have to prep the images as much as you would for archiving. You can give them as they are. And finally, for large data sets that are over several gigabytes, such as is my case, for example, you should prepare minimally sized sample data set for others to test your workflow and trust your process. So where do we publish this original image data? On public data servers. So when you have a ready workflow package, you usually have 2 classical forms of submission for it. And one is as a supplementary material to your main paper. And the second one is as a standalone method paper. For example, in the F1000 Research New Bias Gateway, which is a gateway by the New Bias, the bioimage analysis network on the journal F1000, that allows you to publish your code as an article itself. Like, it doesn't need anything else. It doesn't need to be about the experiment or about the actual data that you got. It's a way for you to publish a methods paper with your code. It's pretty cool. And you can also publish it as a preprint in bioRxiv. In any case, these two classical forms are text formats, so the workflow package itself should be shared on a publicly accessible location, such as GitHub, Zenodo, or SourceForge. Speaking of, GitHub plus Zenodo is, according to the authors, the killer combo. And the reason for that is that it gives you a DOI for your workflow package, which is permanent, and which allows you to have stable links for your main paper with access to this package. And I tested the author's instruction to set it up, and it's indeed super easy. You can follow the instructions, it's super simple. So finally, I want to just mention a recommendation that they made for editors. And as, like I said, there is no simple set of guidelines or rules that editors can implement and that it will fit, that will be useful and fit everybody out there. So what the authors offer is that as when you're about to publish an article, you do self-evaluation of your image analysis reproducibility level. And they offer a table with 3 levels of reproducibility that you can use to classify your work for each element of the workflow package, basically. And it looks like that. So you have your elements can be fully reproducible, which is the scientific approach, meaning that, for example, the description of your code is you have the complete workflow package, the hardware used is provided, instructions are given somewhere like in the manuscript or on a separate resource. Your software, the software I use is available, open source, the data is made available, and the code is directly available online. So this, if all of your elements are there, you're fully reproducible. You have the largely reproducible approach. It's like, well done, but could do better. For example, if you used software that may be commercial or some of the data is available but upon request or things like that. And then you have my favorite approach, which is what I feel like I'm reading all the time when reading articles, is the trust me, I'm a scientist approach, where people basically tell you, oh, I've analyzed this with that software. And that's it. So everything other. I counted stuff with BG. Thanks. So yeah, like these unfortunately very frequent thing is like people do not provide anything, nor the data, nor the software that they use, or the software is not available, and the code does not exist, or they don't give it, for example. So that's it for the article. I give you in the references that I used, and I created a bunch of useful resources for reproducibility and image analysis. You have cool websites that store courses, article guidelines, stuff like that. Encore also provided an example of a journal's editorial policy for image integrity and standards, the one from Nature. And I also put my favorite articles for when you're a beginner in bioimage analysis and you want to know a little bit more on how to be reproducible. Thank you.